Welcome to the second part of Chapter 1. This lecture is going to finish up all of Chapter 1. We are going to start with microbiologists or people important to microbiology. The thing that you need to make a note of for each one of these people that we mention is their last name and why it is that they are important. Remember, we haven't had the means to study microorganisms for all that long, unlike being able to study multicellular eukaryotes. So everything we know about microbiology has only come from the past 300 years or so. Now with more modern technology, we are able to study microorganisms substantially better than we have in the past. But again, we haven't been able to do that for all that long. The beginning of microbiology started with Robert Hooke in 1665. He did build the first compound microscope, which is what we primarily use in microbiology, is compound microscopes. He was the first to characterize the structure and name of cells. So he is known as the father of cell theory, which is going to state that the cell is the basic unit of organization of all living things, as well as that cells arise from other pre-existing cells, which is kind of an important concept that we are going to eventually move into. Our other early microbiologists, so these guys are the first two, is Antoine van Leeuwenhoek. In 1674, he is named the father of microbiology due to kind of an accident of his profession. He was a draper, which meant that at that time he sold cloth, and in order to see the threads and thread fibers, he built a microscope. A very strong one, actually. And when he looked into the microscope at his threads, as well as he would look at other things underneath this microscope, he noticed small organisms moving around, and he began to draw them, which there's a picture of one of his drawings, and he called them animalcules. Try and saying that 5 times fast. He sent his drawings to the Royal Society of London, and his drawings are still referenced today in microbiology. What's kind of interesting about Leeuwenhoek is he went a little crazy seeing small microorganisms only under the microscope, but you can't see them with the naked eye. And he began looking at pretty much anything and everything underneath the microscope. And when I say anything and everything, I literally mean everything. So he was the first to document muscle fibers, bacteria, spermatozoa, which is motile sperm, red blood cells, urate crystals, which you find in gout, and he was the first to see blood flow in capillaries. So he's pretty important as far as that documentation of finding microorganisms. Our next important contributor to microbiology is Francesco Redi in 1668. Began looking at experiments of spontaneous generation. So this spontaneous generation is where small living things were believed to just poof into existence because something so small, where could it possibly come from? And this was the believed hypothesis from 1665 until 1859. So Redi ended up questioning where these small life forms actually came from. So he did an experiment using meat, where he put a slab of meat into a jar and left it open versus a slab of meat in a jar that had a lid over the top. And what we know is that uncovered, organisms like flies would land in the meat and end up creating these worms. So maggots, right? Because they're laying their eggs. That would be the uncovered jar. In the covered jar, flies weren't able to land in the meat and plant their eggs, and therefore, if it was a sealed container, then we see that there's no small life forms being formed because they can't access that product. So this is what Reddy showed, and yes, he was pretty well supported that spontaneous generation didn't happen with things like worms and maggots and things like that, but microbes would be the exception. That microorganisms like bacteria and things like that, they probably just pop out of existence because they are just so small. So let's talk about our next contributor and how they questioned that theory. Our next microbiologist in the making is still going to be during that time that we believe in spontaneous generation. This is Lazzaro Spallanzani. In 1745, he was an Italian priest and professor, and at this point in time, people did commonly know that boiling a product or food would kill microbes. So what Spallanzani did is he boiled meat and vegetable broth to make essentially a gravy for long periods of time, and then would seal the neck of the container by melting the glass. At this point, he had both an open glass and then a sealed glass of boiled gravy. And what he found was that the gravy that had the open flask would be growing all kinds of microorganisms from mold to bacteria, while the sealed flask was very free of microorganisms, which ended up leading to the hypothesis that microbes need air to grow, and that was why the gravy in the sealed flask did not grow any microorganisms. This then leads us to our end of spontaneous generation belief time period in 1859 with Louis Pasteur, which is probably a name that you're at least maybe a little bit familiar with. He did an experiment where he placed nutrient broth in a swan-necked flask, which meant that there was still the ability for air to get into the flask. He then boiled the nutrient broth and then let the nutrient broth in the flask with the swan neck sit for roughly a year. And in that amount of time, there was no microorganisms growing inside of that flask, which is really interesting. So as soon as he broke the swan neck off and had an open line directly into the flask, it then began to grow microorganisms. What this did is help support that microbes do not form through spontaneous generation, even if the flask is open to airflow, which is going to lead to our next big belief that biogenesis exists. And this is going to be where living cells can arise only from pre-existing living cells. So this is that second part of cell theory that is important. So life makes life is essentially what this means. And remember that when we talk about bio, B-I-O, that means life, and genesis means to make or create. So we are saying that this is the creation of life. Now, Louis was not just a one-trick pony. He is noted for a handful of other microbiology advancements. But before we jump into that, I do want to say he did start out as a portrait painter and became a teacher before he started to study chemistry as just like a hobby. He was a professor at several French universities before he started working heavily in microbiology. So at one point in 1857, he was approached by several French merchants to figure out why their wine and beer was spoiling when it was being shipped overseas. So he set out to figure out what was happening. And what he found is kind of the whole process of fermentation where yeast, in the absence of air, converts sugar into alcohol, which is that whole process of fermentation. Upon further discovery, he ended up finding what was causing the spoiling of those beverages was the presence of bacteria that would convert that alcohol that was converted from sugar by the yeast into acetic acid, which is essentially vinegar. So that is what was happening. Now, kind of as a little side note, alcohol isn't a new thing, and I'm not saying that Louis Pasteur discovered fermentation. The thing that he did figure out is documenting why it happens, how we get alcohol. But for the record, humans have been getting sloshed for up to 100,000 years. That's a third of our time as a species on this planet, because we've been around for about 300,000 years, give or take a bit. So about a third of that, we have had the ability to make alcohol. Now, fermentation is not the only process that Louis kind of figured out. He also was looking at pasteurization, which is something that we use today. This is a solution to other products other than alcohol spoiling. This is used primarily maybe for dairy products, milk, as well as for fruit juices. So it's this process of killing microbes within those liquids using heat for up to a certain amount of time, roughly 30 minutes, with no oxygen. So what this would do is kill the microbes that would cause the spoiling and souring of these products in order to make them more shelf stable. So that is a practice that we use now in our dairy products as well as fruit juices. All right, scooting right along into our next scientist, and that is Dmitry Ivanovsky, sometimes spelled Iwanovsky, in 1892. Now, his big claim to fame is discovering, and I'm going to say kind of in quotes, the first virus. Now, what's neat about him discovering this first virus is that he was actually a botanist by trade, and there was some type of disease going around with his tobacco plants. What he did is he took the diseased tobacco leaves, ground them up, and then extracted the liquids from that ground up set of diseased leaves, and then he would spray healthy tobacco plants with that filtrate. From there, the tobacco plants that he sprayed would become diseased as well. So he found some disease-causing agent that was smaller than any microbe at that time being able to see or notice something. So he ended up having to be able to see it with an electron microscope. But again, viruses have been around for plenty of years prior to 1892. It is that Ivanovsky essentially figured out and documented that it wasn't a microbe that had previously been documented causing this disease of his tobacco plants. That leads us to advancements in medical microbiology. So we're going to talk about this term, aseptic technique. This is going to be the use of sterile techniques to prevent contamination. The first thing that should pop into your mind as far as an aseptic technique should be immediately washing your hands. That is going to be the first surefire way of making sure that what you're using to examine patients is going to not contain any contaminants. So, our first guy we need to talk about is Joseph Lister in the early 1800s. He observed doctors that washed their hands more frequently or at all had more of their patients surviving during surgery. And I know you're thinking, well, duh, but keep in mind that microbes, and especially contaminating one person to the next with microbes as a doctor, was kind of a not a crazy concept, but it wasn't something they thought about even slightly, not like we do today. Our next two individuals had kind of a similar discovery, and around the same time in the mid 1800s, we have Oliver Wendell Holmes and Ignaz Semmelweis. Holmes noted that women who had home births would contract fewer infections, which is kind of an important thing to make a note of because women who didn't go to the hospital where doctors were barely washing their hands, everything's contaminated, maybe there's dirty bed sheets and all kinds of things, they would contract diseases from the hospital itself versus just staying home and having your baby. Semmelweis noted that doctors that went straight from an autopsy to live patients, particularly those pregnant women who came into the hospital, had a higher rate of infection and likely also higher mortality rate. So, these guys were some of the first advancements in medical microbiology to start noticing and possibly, hopefully, encouraging better aseptic techniques. Our next individual is Ferdinand Kohn in the 1870s. He discovered heat-resistant bacterial endospores that could survive heat treatments. So what these spores are kind of like bacteria making something like an egg, but it's not really an egg, it's a spore. And what's interesting is that these are very difficult to kill or sterilize, and that heat was the main way in order to sterilize things prior. But after Ferdinand discovered these spores, he realized that we now have to use something more to kill it. Now what we do is we use heat and pressure, and sometimes even then the spores will survive. But what that heat and pressure does, like with a machine like an autoclave, which is a gigantic pressure cooker, essentially, it will explode the spores. So these would have been very difficult to kill, especially in the 1870s at that time. But at least we were starting to figure out that some bacteria and products of bacteria were very difficult to kill. Next up is Edward Jenner in 1796. You'll notice that our dates kind of jump back and forth just as we move through these, so I would not be keeping notes as far as the dates go, but more by the person and know the date, right? What Jenner is known for is being called the father of immunology. Now, his big claim to fame is noticing a link between individuals who contracted cowpox would not contract, or if they did, it would be very mild if they contracted smallpox. Now, you've probably heard of smallpox. You may not have heard of cowpox, but cowpox is very similar in its representation to smallpox, except smallpox is incredibly fatal, especially at this time when there was no vaccine or treatment for that condition. At that time, smallpox killed roughly 10% of the global population and would have killed more if Jenner did not figure out essentially one of the first vaccines. Unfortunately, he kind of went about it in a not so great way. He collected the pus from the blisters caused by cowpox and would inoculate people and then expose them to smallpox to see if they would contract it. The first individual that he did this with was an 8-year-old boy, and it was successful. So then he did it 23 more times, including testing out his hypothesis on his 11-month-old son. So while, yes, he did come across a really fantastic discovery, what if he'd been wrong? That would have been really bad, and he probably would have killed quite a few people, including an 8-year-old child and his 11th-month-old. So it just happened that it worked out well for Jenner. Some cases, that's not the case. Another individual who we know and love is Louis Pasteur. He also created a vaccine to something really important. So he studied rabies in rabbits. And at one point in time, he was approached by a father whose son had been bit by what was assumed to be a rabid dog. So a dog that was diseased with rabies. And at that time, that would have essentially been a death sentence for this child, who was a 9-year-old boy named Joseph Meister. Baby Joey was pretty lucky in coming across Louis because of his research in these rabbits. What Louis did is he mashed up rabbit brains and spinal tissue that he had previously infected with rabies, let it age, let it dry, and then what this did to the virus is made it less powerful, less virulent, and then he injected Joseph, who had already been bit by this dog. And what happened is 9-year-old Joseph ended up living until he was 64 years old. He did not die from the vaccine, nor did he die from the dog bite, and he went on to work at the Pasteur Institution in France until the day he died. Our next individual takes us into 1876 with a German physician named Robert Koch. Koch was developing methods for growing individual types of organisms in the lab so that they could be studied separately, and he then used these methods to examine the relationship between microorganisms and the various diseases that they can cause. Through these studies, he and a colleague ended up developing something called germ theory of disease. This is going to state that a specific organism causes a specific disease in humans or animals or whatever it was we were studying at the time. Koch specifically studied anthrax, which was an extremely deadly disease of cattle and sheep at that time. He noticed a few things, and when we talk about his observations, what we call these are Koch's postulates. They're listed out here on this slide. Please make sure that you know them. The first postulate is that microbes must be present in every case of the disease. The second is that microbes must be isolated and grown in culture from a patient with the disease. The third is that the disease must be caused in a new patient from the grown sample. And the fourth is that microbes must be recovered from the newly infected host. The image we see here is going to be a representation of those 4 postulates I mentioned on the last slide, and you're welcome to use this image to help you study or help better understand Koch's postulates. All right, that takes us into the 1900s. from Koch's postulates, we have two individuals who started looking at basically that we know microbes exist and can cause diseases, but then how do we stop that attack? How do we fight microbes? How do you fight something you can't see? So that leads us to antimicrobial chemotherapy. So when we think of the word chemotherapy, I know your brain immediately probably goes to cancer treatments, but gentle reminder that chemotherapy is just meaning chemical uses to combat something, specifically like diseases, right? So we have Paul Ehrlich in 1910 who discovered the first synthetic drug, which he called the magic bullet. These were small doses of arsenic, which was to treat against a particular sexually transmitted disease known as syphilis. So it was actually really effective in fighting syphilis because if you do not know the results of syphilis, they can result in sores, blindness, insanity, and then death. So to be able to treat that condition or that disease was pretty big. Granted, we are using arsenic to fight that, and we don't use that anymore, but hey, gotta start somewhere, right? From there, we move on to Alexander Fleming in 1928. Alexander Fleming discovered the first antibiotic by looking at the mold Penicillium and then naming that antibiotic after penicillium, which is penicillin. So that was a really, really big discovery from Alexander Fleming in 1928. Big one. I'd put a huge star next to this guy as well as his discovery. We will talk about antibiotics more and how even though that was a fantastic discovery, bacteria ended up responding to penicillin pretty quickly once we started using it. But we'll get back to that eventually. As recently as 1970, we ended up finding the discovery of restriction enzymes through these three individuals, Daniel Nathans, Werner Arber, and Hamilton Smith, where, if you remember from the first part of Chapter 1, we've talked about this with genetic engineering, that restriction enzymes are like molecular little scissors found in bacteria as part of their immune system, and it can be used to chop up DNA in specific locations. This ended up becoming this massive revolution in biological processes in the 21st century due to these three individuals finding or discovering restriction enzymes in bacteria. And then we stole that process essentially from bacteria to be able to use now to treat previously untreatable diseases, which is kind of phenomenal, crazy cool. I have a lot of love for this individual. That is Kary Mullis. He invented the technique known as PCR, which is polymerase chain reaction. And what this process allows is for the detection of small amounts of DNA and then be able to amplify those pieces in large enough quantities that we can use them for studying or in some instances in crime scene investigations. So if there's only a small amount of DNA, you need to amplify it. It's thanks to this guy, which is super cool. He ended up winning the Nobel Prize for this in 1993, which again, I kind of have a little bit more love for this guy. One, he's adorable. Look at that smile. And two, 1993 was the year I was born. So, of course, I feel kind of a strong connection to this gentleman. Also for his crazy, amazing advancement in biology. Now, that does mostly end the individuals that you need to know for this chapter, but we will talk about two more people here at the end of this lecture that you still need to know. But for the most part, that's kind of it for the moment. We are going to switch gears and start talking about RNA, which is kind of similar to DNA if you remember this from previous classes. Remember that the central dogma or that central theme of biology is that DNA goes through transcription, which is a copying process, and is converted into RNA. RNA is then going to go through translation and be translated into a protein. So we go from DNA, which is going to be our A's, T's, C's, and G's, to RNA, which is A, U, C, and G, and then into a protein, which is going to be that physical structure that we're talking about when it comes to organisms in general, right? So what we're seeing is potentially that the central dogma might not be true. It might actually be a little bit of BS. So it turns out that not all DNA leads to RNA, and not all of that RNA ends up leading up to proteins, only about 2%. So if it is such a small percentage that actually goes through this central idea around biology, then what are we actually looking at? What does that actual central theme mean if it's not potentially true? So these small pieces of leftover RNA help regulate what happens in our cells, but we still don't really understand what all is going on. The big thing here, guys, that I want to talk about is one, this is recent information, so like early 2000s, this was being figured out, and it's still in the works, right? The other thing I want to point out is science is always self-correcting, and remember, it's okay to be wrong as long as we learn from it. So if by the time you are, I don't know, nice and old, something like that, you're finding out that that central theme of biology is actually something else, just remember this lecture, however many years from now, that we started telling you about it now. Another advancement in microbiology centers around a program that I have already told you about, and that is the Human Microbiome Project. This is going to be a massive study of what microbes live in and on humans, as well as maybe links to diseases based on those microbes. So what you can do is you can donate samples from all parts of your body, essentially, to the Human Microbiome Project, and then they do all the research and figure out all that comes with those microbes on and in our bodies, which is a really interesting project. That then moves us into the superstructures of life known as macromolecules. So macro in macromolecules means big molecules, and these are the four big ones. You've probably already heard of them. They are carbohydrates, lipids, proteins, and nucleic acids. So all organisms are going to be made up from these few major types of biological molecules, and we are going to talk about each one individually. Our first macromolecule is going to be carbohydrates. I really want you to highlight, underline, star next to where we have the uses, which is energy, and then cell structure. So it's an energy source. It's also an energy storage. The main monomer within carbohydrates is a monosaccharide. Mono means small, which this is just translating into that it's a simple sugar like glucose. Sucrose is going to be another type of carbohydrate, so like table sugar. And then we have starch and glycogen. Starch is going to be the carbohydrate storage system in plants, and glycogen is going to be the storage the main energy storage within animals, so including us. The next type of macromolecule is going to be lipids, and what you probably associate with lipid is probably fat. This macromolecule is similar to fats. Keep in mind that fat stores more energy per pound than carbs, but your body is going to burn carbs before it burns fat, which is why it can be so difficult to drop a few pounds. This is also the idea behind the keto diet, where you massively limit your carbohydrate intake, your sugar intake, and you only consume fat so that your body switches gears and it goes, oh, I don't have any carbs to burn. I am going to burn nothing but fat, and therefore you lose weight. However, with most diets that are restrictive like that, the second you get off that diet, you will start putting back on the pounds pretty quickly. So when it comes to lipids, our uses are energy storage, insulation. You have to have fat, guys, by the way. If you're ever like, oh, I need to get rid of every little shrapnel of fat on my body. No, no, no. You need it. It's going to protect your organs. It's as well as going to keep you warm. It is protection. It acts as a cushion, which is important for many parts of your body, including your kidneys. It creates steroids and waxes, which we are going to talk about, and it is the building block for sex hormones. We're also going to talk a little bit about hormones as well. The monomers are fatty acids plus glycerol. And lipids are typically going to fall into being hydrophobic, meaning that they are water-fearing. They repel, it repels water, which is going to be important in the next thing that we talk about. Most cells are going to have a cell membrane that is composed of a phospholipid bilayer, where the outside of our phospholipid bilayer is going to be a polar head, and then the inside of that layer is going to be a nonpolar hydrophobic tail. So the way that these are structured, and I want you to look at that image in the upper right quadrant here, where we have water on either side of that phospholipid bilayer, and the tails are meeting in the middle, where the heads are then on the outside. This is going to cause a semi-permeable membrane, meaning that only certain things can pass through this phospholipid bilayer or exit the cell, because this entire double layered situation is going to go all the way around and encompass our entire cell. Keeps things in, keeps things out. It's a good system. All right, still talking about lipids. I said we talk a bit about steroids. Steroids can be found in animal hormones, as well as, specifically our sex hormones. This is going to be part of the cell membrane. And you can have examples of steroids within the body, such as cholesterol. That is the base for other steroids and found in the cell membrane to help support the membrane. Cholesterol can come in two flavors, either bad cholesterol, which we call LDL, or low density lipoprotein. This is kind of the unhealthy, leads to heart disease. Examples we can think of as far as things that we consume that cause this really bad cholesterol to build up is butter, as well as red meats. What this can cause is a narrowing of the arteries due to a plaque buildup, and this can cause clots and blocked arteries, heart attacks, possibly a stroke. The way to combat bad cholesterol is through HDL, or good cholesterol. This is high density lipoprotein. This is very heart healthy foods. Good examples include avocado, chicken, salmon, many various types of nuts like walnuts. Red wine is also considered an HDL. Cholesterol, yogurt, oatmeal, blueberries. There's quite a few examples of good cholesterol foods that basically act like a sandpaper through your arteries that then break apart and scrape away at the plaque that is built up from the bad cholesterol. Sometimes we come across things like these waxes that cause certain microbes that produce the waxes to be able to repel lab stains, which makes it harder for us to detect those microbes when trying to diagnose an individual coming to you with certain symptoms. Now, these waxes are kind of interesting in that, much like a wax that you might think about from like maybe a candle, they are pliable and soft when warm. So think about like a lit candle, how the wax becomes very pliable, sometimes liquidy. But when it is cold, it becomes hard and water resistant, much like candle wax, if you think of it that way. So materials like fur, feathers, fruits, leaves, insect exoskeletons, and even human skin are naturally waterproofed with a coating of these waxes. Moving on from lipids, we are moving into our third macromolecule, which is proteins. The uses for proteins are enzymes, antibodies, cell markers, structural material, ribosomes, transport channels across the cell membrane. Honestly, the list kind of goes on when it comes to proteins. Proteins are such a massive part of our macromolecules and make up so many parts of organisms. Everything is pretty much a protein, if you want to think of it that way. The monomer that you need to know is amino acids. And if you remember transcription translation, remember that during translation, we are translating those amino acids into, or I'm sorry, we are translating RNA into the amino acids, right? And then the amino acids end up building into a chain, which creates our proteins. In building those proteins from the amino acids, the proteins are going to have specific shapes. This is super duper important. Please underline, highlight that their shape is going to be related to their function. Much like a key to a lock, this is a very important thing to make note of. Proteins are going to be built a specific way and have a specific shape for a specific job or function within our bodies. And if that shape is changed at all, we call this denaturing. So if a protein is denatured, and this can happen through heat, radiation, pH is thrown off, electricity, chemicals, then that protein cannot perform its job. It is no longer going to be a key that fits that specific lock. It has been deformed. Last but certainly not least, we have nucleic acids as our final macromolecule. These have the use of DNA, which is deoxyribonucleic acid. Something that you should remember is the genetic coding within our chromosomes. RNA, or ribonucleic acid, which is going to be used for protein synthesis in that process of DNA to RNA to proteins. And remember that the word synthesis just means to make, so we're just saying making proteins. And then ATP and ADP, which you can write next to these guys. It's going to have everything to do with energy. So we'll talk about that here in just a second. Now, the monomers include nucleotides, and nucleotides are going to be made up of sugar, phosphate, and a nitrogen base. We are going to talk about these a little bit more on the next slide as we talk about DNA. All right, moving on to DNA. DNA is going to be stored in the nucleus of eukaryotic cells. Remember that eukaryotes, the true nucleus, are going to contain a nucleus. They are the only types of cells that do. The DNA is going to be stored there, housed there, situated there, however you want to think of it. In prokaryotic cells, DNA is not housed in a nucleus. It's just kind of free floating around within the cell. And there is a big difference between prokaryotic and eukaryotic DNA in that in humans, we have 46 chromosomes, and specifically they are linear. So they are kind of like segments or lines of DNA all kind of mushed together like a bowl of spaghetti inside of the nucleus. Meanwhile, in prokaryotes, the DNA is going to be one single chromosome and it is circular. Okay, it's going to be like a ring or a hoop, and they only have one. Very important thing, important distinction to make. We have this term gene, which is going to be a particular segment of DNA that codes for a particular trait. We're going to use terms like genotype and phenotype eventually, and this is a good time to kind of mention it. So a genotype is that specific genetic coding for a phenotype, which is going to be the physical display made with the proteins that DNA codes for once we turn it into RNA. Now, gentle reminder that our nitrogen bases when it comes to DNA is guanine, cytosine, adenine, and thymine. Now, thymine is highlighted because it has a special little side buddy that it turns into, or not that it turns into, but that takes its place is a better way to say it when it comes to RNA. In RNA, we do not have thymine. It is going to be uracil. So guanine pairs with cytosine. That is the same in DNA, same in RNA. Adenine will pair with thymine in DNA, but in RNA, adenine is going to pair with uracil. So RNA is also going to be found in the nucleus as well as in the cytoplasm. So it just depends on what's happening within the cell as far as where the RNA is going to be. Remember that RNA is going to be made from DNA through transcription, right? And that means that through that copying process, we go from DNA, which is double-stranded, to RNA, which is single-stranded. Now, I am going to kind of summarize a lot of what I just talked about with this slide, but I do want you to make sure that you take notes over what precisely we're seeing. So on the middle left, we see DNA, which, make a note of it, is double-stranded. Okay, our base pairs are going to be adenine pairing with thymine, thymine pairing with adenine, cytosine pairing with guanine, and guanine pairing with cytosine. Again, it is a double-stranded helix. Okay, very important. The outside of that helix is going to be sugar phosphates that go back and forth, and we'll talk about transcription translation a little bit later and come back to that information. With RNA, after DNA has been transcribed, it has been copied, we get RNA. RNA is going to be, make sure you write it down, single-stranded. The other important thing is that adenine is going to pair with uracil instead of thymine. So when we make that copy and we see an adenine, then the RNA is going to make a uracil in the place of thymine during that copying process. Cytosine and guanine will still pair up the same way that it did in DNA, but again, we're only seeing half of that strand. That now takes us to the third important part of nucleic acids. So we talk about DNA and RNA, and now we're moving on to ATP and then eventually ADP. ATP is going to be an important energy carrying molecule known as adenosine triphosphate, but you can just know it as ATP, which is perfectly fine. It's made of adenine, a ribose sugar, and the big important part of this structure, which is the three phosphates. So you can see them in yellow on the left on the bottom. Those three phosphate groups, super duper important. Now, how this structure carries energy is that it has a negative charge, meaning that we have a lot of high tension in the bonding for those phosphates to get away from each other. And then when one does pop off, what that becomes is ADP, and that actual process of removing that phosphate group happens with such a force that it creates the energy that is ATP, that ATP is known for. Now, we will eventually talk about cellular respiration, which is going to go through this very big process of ATP going to ADP, going back to ATP. Now, the reason that this cycle happens in this way, that we cycle through ATP, ADP, ATP, and so on and so forth, is it's not dissimilar, this molecule, to a cell phone. If your cell phone loses all of its energy and the battery dies, you don't just throw the phone away. What you do is charge it, which is precisely what this molecule is going to do. It's the same kind of concept. So when we get to cellular respiration, kind of keep that in mind, and we are going to go more in depth in a later chapter, but just keep that in mind for now. So that kind of does it for our 4 macromolecules. Remember that collectively these guys are going to combine to form cells. Some living things like bacteria will only consist of a single cell, whereas other organisms, mainly eukaryotic organisms like plants and animals, are going to be made up of trillions of cells that are composed of the 4 components we just talked about, those 4 macromolecules. Speaking of cells making living organisms, well, we have to talk about how all of these different organisms are classified, identified, and what that leads us to is the overall arching term taxonomy. So with all of the diversity that we find on Earth, we need a way to classify all of it. And that's where this taxonomy comes in. So created over 250 years ago, here's our next guy we gotta know, is Carl Linnaeus, or Carol Linnaeus. He ended up creating a set of very basic rules in order to classify and establish taxonomic categories, which we call taxa. This is going to be important because it's going to help us discover what it is we're looking at, what it is we're studying, and what category they fall into. So if I had to go in order of this taxonomy, the first thing we do is identification of an organism based on observations of their traits in order to be able to be recognized and then named and then placed in our overall taxonomic system. From there, we would go into the classification, which is that bottom term, which is going to be the arrangement of our organisms into a hierarchy. And we'll talk about that hierarchy here in just a second. I'm sure you've probably actually heard it before, but we are going to go over it. And then finally ending with nomenclature, which is going to be assigning a scientific name to our taxonomic categories and individual organisms. So this is going to be sort of the difference between a common name versus that Latin scientific name. So common names are going to be things that we're all pretty familiar with, like a robin or an earthworm, black-eyed Susan, groundhog, and the list goes on. Carol Linnaeus ended up establishing a naming system that is going to be binomial as well as mainly based in Latin. And we'll talk about why it needed to be Latin here in just a minute, but give me a second on that. So this binomial system is going to be a two-parter system. So bi meaning two. You can kind of think of these scientific names as being like a first and a last name. Like if I gave you guys my first name, which is Rachel, that would be my genus, if you want to think of it that way. And you can't really get as much information about me just from Rachel. However, if I give you my last name, which is Sullivan, that would be my specific epithet, then you know that my full name is Rachel Sullivan, which can really help you in identifying and classifying me, I suppose. So when it comes to our genus and our specific epithet that creates our scientific names, for humans, it's Homo and sapien. This translates into thinking or wise man. If we just stopped at Homo, the genus, then we kind of don't know as much about that specific species. That's the important part because we are not the only organisms in the genus Homo, which is why we have to add sapien. That tells us that we are specifically the most current species of human. But we weren't always. There was Homo erectus, Homo neanderthalensis. There have been a few. So that lets us know through the genus and the specific epithet what organism within that hierarchy we are specifically looking at. Now, you probably noticed on that last slide that Homo sapien was italicized and the H in Homo was capitalized. And you can see this at the bottom of this slide here. It is important for you to note how to specifically write out scientific names. Please make sure you highlight this, put a star next to it, something. All scientific names are either italicized if it is typed out, or if you are handwriting it, it needs to be underlined. The genus needs to be capitalized, and the specific epithet can be in lowercase. It should be lowercase. This is the proper way to write out a scientific name. This is going to be really important once we get to specific species that we're working with in lab. You will have to make sure that you write out the scientific names properly, or I will take off points. When it comes to scientific papers or just papers in general, after you write the scientific name the first time properly, if it's typed, italicized, if it's handwritten, make sure you underline it. From that point on, you can kind of shorthand the scientific name where you just use the first letter of the genus and then put a period. And then you can write out maybe a shortened version, or it's not usually a shortened version. I don't know why I said that. You can write out the second part of the name, that specific epithet, such as Staphylococcus aureus. You would shorten the Staphylococcus just to S and then just use aureus after that. Now, you might be wondering where these names even come from, as far as the scientific names, and a lot of it is going to have to do with the person who found it, or maybe a description of the organism or where it was found. For example, Staphylococcus aureus, it describes a clustered arrangement of cells, staphylo meaning a bunch of grapes, and the golden color of the colonies, or ore, means gold. So it's basically just describing what that bacteria looks like. Another example is E. coli. It honors the discoverer, which was Theodore Escherich, as well as describing the bacterium's habitat, which is the large intestine or colon, and that is where you get the term coli in that bacteria's name. Now, why does it have to be Latin? So, fun fact, Latin is a universal language. It's also a dead language, which both create a fair playing field that means that that language will never change and it'll never have slang. So if you think about the name polar bear, is a pretty good example. In Spain, they call it oso polar. In Germany, they call it Eisbar. And then in English, we call it a polar bear. So there is not a common term for polar bear except for Ursus martimus, which is the scientific name, and it is the same in every country that is the scientific name. You can think of kind of in terms of English, even in all English speaking countries, there are certain terms that mean different things. Like in America, we say escalator or elevator, and across the pond, they say lift. We mean the same thing, but it's a different term for that one item, right? Not to mention the constantly changing terms and slang for different words. This can be very confusing when it comes to talking about identifying organisms, which is why we have this unifying language that is a dead language that will never change, that is Latin, and why we use Latin in our scientific naming. Now we have this overall classification of organisms in the form of this hierarchy. It's a descending rank system, essentially, starting with domain, ending with species. Domain is going to be the most broad and inclusive of all of the taxonomic categories. And then, of course, we get incredibly specific with species. You do not need to know all the different phylums, all the different classes, orders, etc., but you do need to know the order of domain, kingdom, phylum, class, order, family, genus, and ending with species. So, the only one of these that you have to know what it includes is domain. Remember, there are three. We have bacteria, archaea, and eukarya. That's the only one of these ranks that I'm going to ask you to remember the parts of. Otherwise, you just need to remember the order that these classifications go in. You can remember it in the form of maybe an acronym like DKPCOFGS, or you can remember it with like a little term, like a sentence, like Dear King Philip climbed over the fence for good soup. Something like that might help. So despite what I've shown you before, it's actually an updated version of the Linnaean system of hierarchy, and now we use the Woese-Fox system. This is the more updated version from Carol Linnaeus's classification system. The big thing that I want you to get here is just that we've updated it prior to this. We did not have archaea separate from bacteria, but now we have it separated. So we have eukarya, bacteria, and archaea. They are no longer together. And also previously, Linnaeus had 5 kingdoms that divided all organisms. But upon further information, that led to the addition of those 3 domains that I just mentioned. So we just have an updated format, and that is the Woese-Fox system. Now the last thing we're going to end on is that gentle reminder what we use this classification system for is to build that tree of life that I showed you in the last part of chapter 1, or in the first part of chapter 1. This is going to include all organisms on the planet and ideally all the organisms that have ever been on the planet, minus viruses because we don't necessarily consider them alive. And remember that our tree of life is going to be a work in progress, and it's still particularly difficult to form due to a lot of organisms who have already previously gone extinct. And that is it, folks. That is it for chapter 1. Keep in mind that this was the second part of chapter 1. We have now finished it, and you can go ahead and study on up.